name is Johannes, and I'm going to present an approach for training deep generative neural networks to engineer functional DNA sequences that are high fitness and diverse. And so the problem we consider here is to computationally generate and design DNA sequences that conform to some target function. So very similar to the objective that Jennifer Linder talked about in her amazing presentation. And there are a number of different computational methods that address this particular problem. And they all sort of are based around this idea that you have a pre-trained predictor oracle model that, given an input DNA sequence, can predict this target function of interest. So then, for example, you can apply different search algorithms, like genetic algorithms, to iteratively refine your DNA sequence for your target function. There's also been some really cool work on what's called like feedback GANs for iteratively refining a GAN to be optimized for your function. And then, as we heard in Jennifer Lister's talk, there's been some amazing advancements in adaptive sampling techniques for protein engineering. Here we're going to take a slightly skewed approach. We're going to assume that our predictor oracles are always differentiable all the way down to the input layer, which allows us to use backpropagation as a mechanism for training the DNA sequence to get better at this target function. So the basic principle of generation by backpropagation is that you start with some randomly initialized matrix of nucleotide logits. You then feed this through some kind of activation layer, for example, like a softmax layer, to get your sequence PWM. You then feed some input representation of this PWM to your trained predictor model. In our work, we typically sort of pass this through a sampling layer where you pull actual discrete samples from this PWM. And as far as differentiability goes, we typically pass through the straight-through approximated gradient. Anyway, you use your predictor model to predict the phenotype or the target function, after which you evaluate the objective that you set up, that you want to maximize for. And then, because this entire scheme is differentiable, you're able to backpropagate the gradient of the objective function with respect to the nucleotide logits, take a small step in the direction of the gradient to slowly but surely optimize the logits for your objective function. And there's been, so in earlier work, we sort of used this scheme to engineer functional polyadenylation signals. And there's also been some really cool related work by Killeron in 2017 that essentially did this scheme, but instead of doing backpropagation directly on one particular sequence instance, essentially did backpropagation through a pre-trained generative adversarial network and optimized the latent input seed to the GAN to have the corresponding generated sequence be optimized for this target function. So now if we take a step back, and I think Jennifer Lister also said this, that typically we're interested in generating lots and lots of potential candidate sequences that are all highly diverse, but are all predicted to fulfill this functional target. And so then this generation by backpropagation scheme has a number of downsides, essentially because for every time you want to generate a sequence, you have to reinitialize your nucleotide logit matrix and rerun gradient descent all the way to convergence to get one sequence. Now, if you want to do that for 1 million sequence candidates, you're going to have a lot of computation ahead of you. And also in some optimization runs, you may run into a poor local optima due to the random initialization of the matrix and get a poor sequence out in the end. Finally, you're running this optimization scheme independently for every sequence you want to generate, which means there's actually no guarantee at all that you're going to end up with a diverse set of sequences in the end. So the question we asked was, well, can we replace this single logit matrix for one particular sequence by a generator? By a generative neural network that can predict an entire sequence distribution, and can we then train this generative network, its weights, such that the sequence distribution gets optimized for our target function? The tricky question then is, well, how do we enforce that the generator actually maintains diversity and learns to produce a diverse set of sequences that are all predicted to be functional? So then we came up with this rather simple scheme, which we call deep exploration networks, or DENS. The idea is to jointly maximize the fitness predicted by your predictor oracle, as well as a kind of a Monte Carlo diversity cost term. So at every forward step in this approach, you randomly sample 2 independent latent input seeds that you feed into your generator. You have the generator generate the 2 corresponding sequences. You then feed one of those sequences to your pre-trained predictor model and evaluating your objective function, and then training the weights of the generator to optimize the generator for your functional objective. But then you also compare your two sequences that were generated based on some differentiable similarity metric, and then optimize your generator to minimize this diversity cost. And so you're sort of jointly trying to maximize the functional fitness as well as the diversity of your model. And also note that we start with sort of a randomly initialized generative neural network, and what we're doing is we're training the weights of that generator to minimize this joint cost function of functional fitness and diversity for every possibly sampled latent input seed pair. Just as comparison for the sort of the work architecturally that we build upon by Kiloron, the optimization sort of took place on a pre-trained GAN where the weights were fixed and where the minimization occurs on a single input seed to that model. And so to demonstrate sort of deep exploration networks, we looked at a gene regulatory mechanism that we've sort of studied intensively before called alternative poly-A annihilation. Now, poly-A is something that happens at the 3' end of mRNA, where a sequence regulatory code called the poly-A signal, illustrated here as green and red boxes on the RNA molecule, the sequence code sort of attracts the poly-A machinery, which comes in and cleaves the RNA molecule at that position, resulting in one particular isoform. Now, if you have multiple signals, it means you essentially have multiple possible isoforms for that RNA molecule. And so the design task that we looked at was essentially, can we train our generator to produce sequences that are optimized for different target isoform proportions to essentially engineer APA. And so what we did was that we trained 5 different deep exploration nets to optimize sequences for 5 distinct target ratios. And as our predictor model, we used Apparent, which is a convolutional neural net developed by our group previously. And as far as evaluation goes, we use after having trained the generators, we use them to sample 100,000 new sequences for each of these target ratios, and then essentially just compare what our predictor oracle thinks about these sequences. How well do they conform to the target isoform proportions? And sort of the first result that we sort of observe, the first thing we note, is that this generation scheme is very fast computationally. So once you pay the initial cost of training your generator, you can sample new sequences very fast and generate 100,000 sequences in under a minute, provided you have access to a GPU. The next thing we note is that there's 0% duplication by these 100,000 generated sequences. They're all unique. So the generator seems to learn to produce diverse sequences. We also validated diversity with other metrics such as hexamer entropy and whatnot. And then the third thing we note is that for these 5 different generators, they learn to produce sequences that are predicted to be very close to the target isoform proportions. So then we asked, well, what is really the significance of enforcing diversity by doing this sort of dual training scheme? So then for a separate task, we sort of said, okay, let's try to train a generator to generate maximally strong poly-A signals that result in the strongest possible signal in an isoform proportion. And we first did so with a very small similarity loss coefficient. And surely, the generator learns to produce seemingly strong poly-A signals with a mean isoform odds of 400 to 1. However, it does not learn to generate diverse sequences. So we observe a duplication rate of 99.5% for 100,000 sampled sequences. So only 0.5% of those sequences that we generate after having trained this generator are unique. Now the picture completely changes once we increase this similarity loss coefficient. So now the duplication rate drops to 0, so it learns to produce unique sequences all the time for these 100,000 sequences we sampled. So it's very diverse. And what's kind of interesting to me, at least, is that the mean isoform odds, the functional objective that we're optimizing for, that actually increases rather than decreases. So this constraint of sort of Monte Carlo trying to enforce diversity during training actually increases the functional fitness that we're optimizing for which we suspect has to do with the fact that you're sort of Monte Carlo enforcing that, or promoting the generator to explore during training to generate more optimal sequences. So then, as Jennifer pointed out, just evaluating based on the predictor oracle that you trained on kind of is foolish. So then what we did is we went into the wet lab and we tested some of these sequences. And in fact, in previous work, we had used this basic vanilla gradient descent approach I talked about in the beginning to generate maximally strong polyase signals. Now our predictor said that our new deep exploration network, or DEN sequences, were predicted to be 20-fold stronger, which sounds pretty crazy, but we thought, okay, let's try and synthesize them. So what we did is we took our new DEN-generated polyase signals, the green box there, and synthesized them on a minigene reporter in direct competition with our old gradient descent generated maximally strong polyase signals. And so they are essentially going to compete for getting polyadenylated, and then we essayed the selection odds using qPCR, and we also did it with the reverse orientation to discount for first come, first served bias. And long story short, the odds ratio of improvement that we observed was 11.6. So our newly generated, deeply generated polyase signals were on average 11.6-fold more preferred than our gradient descent generated old polyase signals. And we had previously, in our earlier paper, experimentally tested these gradient descent generated polyase signals, and they were much stronger than typical strong native polyase signals like the BGH PS. And so if we extrapolate, our newly generated polyase signals are indeed extremely strong with a selection odds of 1481 to 1 compared to this BGH polyase signal. So for future direction, we sort of acknowledged that part of our success here was due to the fact that our predictor oracle was extremely robust because it was trained on essentially millions of essentially uniformly randomized polyase signals that we had generated data for with an MPRA. For other sequence predictive tasks or design tasks, predictors may be just terrible on out-of-domain sequence data. And as we all know, sequence space is huge. So what we're sort of thinking about doing next is to incorporate variational autoencoders or GANs to constrain or regularize the optimization of our DENs and borrow ideas from Killeron, essentially. And we're also looking to apply DENs to new design tasks. In our preprint, we actually designed cell-type specific splicing constructs. We're also interested in looking at enhancers and promoters for genes and also protein engineering. With that said, I'd like to say thank you, and thanks to Georg Seely, my PI, Nick Bogart, Alex Rosenberg, colleagues. What I'm showing there is a pretty neat animation of what it looks like when you're training this deep exploration network to maximize the polyase strength. And those are a bunch of sampled sequences during training. It's actually the same seeds over and over, and you can see how the sequences essentially converge and get stronger and stronger. And we also have a web tool up if anyone's interested in looking more at this work. All right, thank you.